Hello, biology. The purpose of this video is to show you the types of questions that you're going to be seeing on this year's spring final. Because this is a video and because you can pause the video and take time to look at your study guide or look things up on the book or look things up on the internet, I won't be spending as much time on these questions or on these slides as I would in class. And what we're going to do is we're going to do one column at a time so you can focus on one area of your study guide at a time. All right, here we go. Oswald Avery's experiment showed that which organic molecule was the transforming factor in bacteria. Remember, in these experiments, he took a heat-killed substance, injected it into mice, and it actually killed the mice. So something was happening with that bacteria. Question answer coming in 5, 4, 3, 2, and 1. So he realized that DNA was the transforming factor. What's the name of DNA's structure? What do we call that twisted ladder structure? And the process by which DNA is copied is called blank blank. Answer coming in 5, 4, 3, 2, and 1. Yeah, so it's a double helix. That's the twisted ladder structure, and DNA is copied in a process called DNA replication. Where is DNA found within eukaryotes? Our cells are eukaryotic cells. Complex organism cells are eukaryotic cells. DNA is a blank made up of many monomers called blanks. The specific location where DNA replication takes place is called the blank blank. Answers coming in 5, 4, 3, 2, 1. All right, so DNA is found in the nucleus in eukaryotic cells. In prokaryotic cells, it just floats in the cytoplasm. DNA is a polymer made up of monomers called nucleotides, and the location where the hydrogen bonds are broken and new nucleotides are added to the growing DNA chain, that is called the replication fork. DNA replication results in 2 molecules. Each molecule has 1 blank strand and 1 blank strand. Which type of mutation can cause changes in every amino acid after that spot, causing the protein possibly to be non-functional? And which mutation is never a frame shift mutation? Answers coming in 5, 4, 3, 2, and 1. All right, so each new DNA molecule has half of the original strand, which is used as the template, and then half of it is the new or complementary strand. Frame shift mutations shift. It's either an insertion, deletion, insertion or a deletion mutation. And what happens is that shifts the reading frame because remember, each amino acid is coded for by 3 nucleotides, and that means that every single amino acid after that spot could be out of sequence, and that could make the protein non-functional. Substitution mutations, they don't remove a nucleotide, they don't add a nucleotide, they just simply change nucleotides. So it's very unlikely that substitution mutations are going to cause serious problems, but there is no frame shift with a substitution mutation. All right, on to genetics. When Mendel crossed true breeding tall plants with true breeding short plants, why were all of the offspring tall? Hint, the genotype of these offspring was big T, little t, because it was big T times little t. That would be true breeding tall and true breeding short. Answer coming in 5, 4, 3, 2, and 1. It's because the tall gene or the tall allele was dominant. Therefore, the phenotype for all of these offspring would have been tall. If a plant has 2 different alleles or genes for the same trait, they are said to be blank. If a black chicken is mated with a white chicken producing a speckled offspring, what is that an example of? Answer coming in 5, 4, 3, 2, and 1. All right, so heterozygous, different alleles or genes. Of course, if it's the same allele or gene, it's homozygous. And codominance, co meaning together. That's when you see both of the dominant traits in the offspring. Luther Burbank used what process to make a new line of plants? Pictures of homologous chromosomes, of autosomes, and of sex chromosomes are all found in a blank. And blank used pea plants to study the inheritance of traits. Answer coming in 5, 4, 3, 2, and 1. Yes, Luther Burbank used hybridization to make new organisms. A karyotype is a picture of all of the chromosomes from a cell, and Gregor Mendel used pea plants to study the inheritance of traits. Mendel is the father of genetics. The law of blank states that one gene from each pair goes to each sex cell. So we're talking about meiosis, we're talking about production of sperm cells and egg cells. The principles of blank can be used to predict the traits of offspring produced by genetic crosses. Most blank genes are located on the X chromosome, and genes that are stronger or mask other gene forms are said to be what? Answers coming in 5, 4, 3, 2, and 1. Okay, so segregation is when the gene forms or the alleles or the homologous chromosomes get separated when gametes are formed, because remember, gametes have to have half of the genetic information. Principal probability helps you predict what's going to happen in a genetic cross. Sex-linked or X-linked genes are located on the X chromosome, and genes that are stronger or mask the recessive form are said to be dominant genes or dominant alleles. The genetic makeup of an organism is its blank. 5, 4, 3, 2, and 1. That's genotype. The physical characteristics, of course, are its phenotype. Which scientist took the famous photo 51 that helped determine the structure of DNA? If two tall plants are crossed, so heterozygous times heterozygous, why are some of the offspring short, homozygous, recessive? Answers coming in 5, 4, 3, 2, and 1. So that was Rosalind Franklin. And what happens when gametes are formed, the alleles or the gene forms segregate. The only way to end up with a little t is if you separate the little t from the big T right here, and that happens when gametes are formed. So the alleles segregate or they separate from one another. Traits that come from the interaction of many genes are called blanks. What scientist or scientists is or are given credit for the idea of natural selection? So is it one scientist or is it two scientists or more scientists, I guess? The sugar and phosphate group form the blank of the DNA molecule. What part of that structure do they form? Answers coming in 5, 4, 3, 2, and 1. All right, so many genes. Polygenic traits are influenced by more than one gene. Both Darwin and Wallace are given credit for natural selection. Remember, Darwin was taking a really long time to go public with his findings, and one of the things that actually got him over the hump was receiving a letter and some information from Wallace, who was doing research in the southern hemisphere. And that basically was the same conclusions or the same findings that Darwin had come up with over his life. And they published, I think it was 8 months after that. They published together, but Wallace always gave Darwin the majority of the credit. And the sugar phosphate makes up the backbone or the two sides of the twisting ladder. All right, on to evolution. Gene shuffling slash crossing over and mutations are the two main sources of blank blank. 5, 4, 3, 2, and 1. They're the main sources of genetic variation, which scientists suggested that too many humans are being born for the available resources we have left. He also suggested that war, famine, and disease will control the population. What Finch trait helped Darwin come up with his ideas on evolution? And I'm going to fix this real quick. Answer coming in 5, 4, 3, 2, and 1. All right, so that was Thomas Malthus, and it was the size or shape of the beak that helped Darwin come up with his ideas. Fix this real quick. A single species that evolves into many different forms is referred to as a blank blank or as blank blank. What's that process called? The percentage of all species that have lived on Earth that are now extinct is about what? One necessary condition for life on Earth for life to form was the presence of what? Answers coming in 5, 4, 3, 2, and 1. All right, so when you have a species that evolves into many different ones, for example, the finches on the Galapagos Islands, called adaptive radiation, over 99% of the living things that have ever been on this planet are now extinct, which hopefully is startling to you and will urge you to preserve what we have left on this planet. And water is important, but liquid water is what we needed for the ability of organisms to evolve. Because obviously, if it's frozen water, they can't move, and it's if in the atmosphere, gravity is going to cause problems. So we needed liquid water, specifically in our oceans. Similarities in finches Darwin observed led him to believe that they all blanked from a common ancestor. All the genes within a population make up a blank blank, and blank blank acts directly on phenotypes. And what type of isolation is necessary for speciation to occur? Answers coming in 5, 4, 3, 2, and 1. Okay, so they descended or they came from common ancestors. The gene pool is all the genes, characteristics within a population. Natural selection acts directly on the characteristics that either give you an advantage and you're selected to survive, or they do not give you an advantage or they give you a disadvantage, and of course you would be selected to die. And reproductive isolation is necessary for speciation, and remember, geographic, temporal, and behavioral isolation can lead to reproductive isolation. I had some more evolution questions and some random ones. The Galapagos, blah, blah. The Galapagos finches are a great example of blank, where one species evolves into many different ones. 5, 4, 3, 2, and 1. Speciation, so where you have one organism and it adaptively radiates out over thousands of generations to different species. That is the process of speciation. Miller and Urey's experiment tried to demonstrate that blank blanks could have existed before life was present on Earth. Bacteria were likely the first types of blank on the planet. Answers coming in 5, 4, 3, 2, 1. All right, so organic molecules specifically, they found some amino acids as a result of their experiment. And of course, amino acids are the building blocks of protein, and proteins are the organic molecules of structure and function. All right. And we think that bacteria were the first life forms, so that would, of course, been our most distant ancestor, if you believe in evolution. Because of the adaptations Darwin observed in finches and tortoises, he wondered if species living on different islands had once been members of the blank species. Darwin observed somewhat blank species with traits that suited their particular blanks. And remember, these questions are harder than the ones on the test. The ones on the test are multiple choice. So if you know the answers to these questions, you should do well on the multiple choice questions. Answers in 5, 4, 3, 2, 1. All right, here we go. So he wondered if they had been members of the same species. Now, I'm not saying that tortoises and finches were members of the same species, but those different animal groups each. He wondered if finches had come from a previous species or tortoises had come from a previous tortoise species. All right, Darwin observes somewhat similar species with traits that suited their particular environments. All right, protein synthesis and some random questions. The process where messenger RNA's code is converted into proteins is called what? Not the process where messenger RNA is made. It's where that code is taken from the nucleus out to the ribosome. What do we call that? 5, 4, 3, 2, 1. It's called translation. Translation is basically synonymous with protein synthesis. Which enzyme makes messenger RNA? Blanks are made by constructing long chains of amino acids. Answers in 5, 4, 3, 2, and 1. All right, so RNA polymerase makes messenger RNA, DNA polymerase makes DNA, and proteins come from the monomers amino acids. Blanks contain instructions for making proteins. According to Darwin's theory of natural selection, organisms that survive have blanks best suited to their environments. Variation of human skin color is an example of what type of trait? Answers coming in 5, 4, 3, 2, and 1. Okay, so genes are instructions for making proteins. Remember, genes are segments of DNA that code for specific proteins. Organisms that survive have characteristics, adaptations, or traits that give them an advantage. And skin color is a polygenic trait, meaning skin color is influenced by many different genes. Deoxyribose, a phosphate group, and guanine make up a nucleotide of a blank molecule. True or false? The instructions for making... Whoops. This is a mistake for making... Sorry about this, guys. Now I'm kind of a perfectionist when it comes to this stuff. All right, let's see if I can copy this over now. All right, sorry about that interruption. The instruction for making proteins are specified by genes. Blank is double-stranded, found only in the nucleus, and has deoxyribose as its sugar. Blank is single-stranded, found at the nucleus and in the cytoplasm, and has ribose as its sugar. Answer coming in 5, 4, 3, 2, 1. All right. Okay, so deoxyribose phosphate group guanine make up DNA. If you would have seen ribose phosphate group and uracil, it would of course be RNA. Yes, the instructions for making proteins are specified by genes. That is true. DNA Deoxyribose, double-stranded, only can be found in the nucleus. RNA, ribose, single-stranded, and it can be found in the nucleus where messenger RNA is made, and in the cytoplasm at the ribosome where proteins are made. All right, y'all, if you feel good about these questions, you're probably ready for the test. If you're not feeling so good about these questions, truly you have some more studying to do. We will see you back in the class.